overall goal of this video article is to provide answers to the following two questions. 1. Can studies of protein interactions be made more reliably by measuring the fraction of a protein sample that is actually capable of binding to another protein? 2. How can kinetic analysis give us a better understanding of structure-function relationships? Both of these questions will be addressed in an experiment involving Biacore X100, an instrument that detects protein interactions in real time without using labels. This experiment will examine the interaction of bovine cystatin B with the cysteine protease Papain. The crystal structure of the two proteins in complex points to several residues involved in binding to the protease. To determine how each of the interacting residues affects the cystatin B Papain interaction, several cystatin B variants are produced via site-directed mutagenesis. In such experiments, there is always the possibility that recombinant proteins will not fold correctly, aggregate, or deteriorate, leading to incorrect and irreproducible results. Biacore X100 has the capability of determining the fraction of protein that is available for binding by calibration-free concentration analysis, abbreviated CFCA. This approach allows for determining concentration of protein with retained binding activity, even when calibration standards are not available. CFCA will be used here to determine the concentration of the cystatin B variants. These data will then be used as input in the determination of the binding kinetics of the cystatin B variants to Papain, which is also assessed using Biacore X100.
Hi, I'm Claes Holmgren from GE Healthcare in Sweden. Today, my colleague Ewa Pol will show you how to do structure-function relationship analysis using Biacore X100. Recombinant proteins may exhibit improper folding or lose binding activity over time, and this behavior cannot be